Welcome to the Genetics Podcast. This episode is part of our Precision Pioneers mini-series, which is focused on people who are on the cutting edge of precision medicine. And I'm here today with Dr. Vineeta Agarwala, who's a general partner at Andreessen Horowitz, where she leads investments for the firm's Bio Fund, including therapeutics, diagnostics, and digital health, with a focus on companies that are using unique datasets to improve drug development and patient care delivery. Since we're talking to an investor today, I just need to note that A16Z investments may be discussed in the conversation, but none of the following content should be taken as investment advice. Please see a16z.com/disclosures for more important information. So Vineeta, I've had the chance to interview quite a few people from biotech companies as part of this mini-series, and I'm really excited to get your viewpoint, both from your investor perspective at A16Z, but also I know you have a lot of direct relevant experience from your time at Flatiron. And also, I understand that you still see patients through some of your work at Stanford, which is great. So I wonder if maybe you could start with a just quick 2 to 3 minute thumbnail history of your career leading up to now, and then we could take it from there.
Sure. And thanks so much for having me on the podcast. I think, in many ways, our industry is turning over, sort of entering a new chapter. And so I feel like I'm just getting started and learning along with a group of really great innovative companies in this space. In short, I grew up the daughter of a physician and someone who was running research computing for a university. And so I very much kind of grew up in a household thinking about where computation could be applied to research and having sort of a natural inclination to think about how it could be applied to help improve patient care. And at some point, in the late 90s, my dad, who ran research computing at Penn State and was constantly orchestrating, access to data storage, data compute resources for different investigators all over the university, at some point said, hey, it used to be the meteorologists and the physicists, kind of using the most computation. And there might be something going on in biology now, because it seems like those labs are really, raising a lot of funds and accessing a lot of that technology to make discoveries in genomics and think about large scale, biological data. And so that kind of, I think turned me on to that possibility and the prospect of, not just academic research, but also companies and translatable science coming out of that ecosystem. And so I kind of have spent the last 20 years on a journey to think through some of those applications and learn about those spaces, really. So I did computational biology research in different areas throughout my undergrad education at Stanford, as well as throughout my PhD and MD PhD training at the Broad Institute and in the Harvard MIT ecosystem, and just have learned a lot along the way, and now have the really unique opportunity to watch companies being formed exactly at that intersection as well, and have the real privilege of supporting a number of companies at that space.
Amazing. Well, thank you. And I noticed just in kind of reviewing your biography that you really started your MD PhD at the Broad Institute in kind of the perfect time. When you look at those graphs of the cost of genome sequencing, I think it's 2007 or 2008 that it departs from Moore's law and goes kind of supernova in terms of how low the cost has gone. So I feel like you couldn't have, it seems like your parent, who is in the IT and data infrastructure side of things, probably had a role to play in that.
Yeah. It was really a lucky time. The more, when I look back on it, it was a fascinating time to be entering the genomics industry. And honestly, my own instincts around where computation was going to play a role in biology before had been a little different. I'd been kind of looking at, gene expression profiling, which was, in the prior 10 years had dominated bioinformatics and computational biology. I'd been looking at, even kind of more hardware-like applications, and worked on a semiconductor-based, PET scan device, right, and thought about image reconstruction. And there's so many places where computation can be used in biology broadly. But just at that time, I met my PhD advisor, David Altshuler, and he said, look, this is going to blow up. If you want to really work on something that's going to be tied to human disease and understanding the biology of human disease, I really think you ought to think about human genetics. And he was so right. It was exactly at that time, 2007, the Wellcome Trust published a number of the very first large-scale genome-wide association studies, not on hundreds of patients, but for the first time on sort of thousands of patients, a genome-wide scan of common variation and actually tying that to annotated disease phenotype was so novel at that time. And then every single year, exactly as you said, it was like, okay, now we can start doing targeted sequencing of a few of our favorite regions across the genome. Okay, now we can start doing, exome sequencing. Now we can start doing it at extraordinary coverage. Oh, by the way, now we learned a bunch of, where the sites of rare variation are, so we can make a chip that includes, those sites of variation and the Omni chip and the exome chip and all these, kind of cost competitive solutions to be able to profile really huge sample sizes came out. And then, of course, low pass whole genome sequencing and ultimately even high coverage whole genome sequencing all became really possible within the course of that short time period of about 7 to 8 years. And I really had a front row seat to watch that play out. So it really was a special time. You're right.
Yeah, I can imagine. I mean, when I was thinking about how to frame this discussion or structure it, I thought we could kind of look at the first 10 years of your, it's a little more than 10 years, maybe 12 or 13, but from when you started your MD PhD to now, and then, obviously the next 10 to 12 years going forward. And it sounds like, what you just said was something that progressed probably extremely quickly. If you were sitting there in 2007 and said, how many people at a time would we be sequencing in 2020? Would the UK have plans to sequence 5 million people, for example? Maybe you would have predicted. I'm curious, generally speaking, what were some of the things that you think in retrospect moved way more quickly than everyone was thinking? And then also on the flip side, what would you have, bet the farm on in 2008 that we'll definitely have this by 2020 and it just hasn't materialized?
Yeah, it's a really good question. I think the recognition that human genetics can play a really key role in prioritizing and discovering novel, drug targets in disease. I don't know that it's necessarily gone far faster than anticipated, but at least I really do think that's become widely accepted. There have been, at this point, even studies that have kind of looked back at drug targets selected by biopharma companies and asked the question that, hey, if there was human genetic data, at the time that a drug program was launched in support of that target as really playing a potentially causal role in human disease, does that drug have a higher chance of ultimately succeeding in clinical development? And the answer to that is yes. And so there's been this kind of real recognition that these are valuable data sets to guide our, discovery of therapeutics that are likely to really move the needle on disease. So that part's been really great to see. That's happened not only via, sort of some high profile partnerships like 23's and, 23andMe partnering their data set up with a pharma company. And, the same has happened with Decode. And now so many companies have created access strategies to large biobank resources like the UK Biobank, like FinGen, a collection of a really interesting set of genome data, in a population that had a historic bottleneck effect to enrich for maybe higher effect size alleles and so on. And so this is happening now. There's more companies than I can count that are incorporating this data into some part of their discovery strategy. So that's been great. On the other hand, where I think, adoption has been a bit slower is probably in the clinical diagnostics realm. And I think part of the reason actually is that we've been hung up on exactly that word, diagnostics. And we've been focused on kind of getting to a mainstream place of utility for human genetic information to play a role in definitively diagnosing disease versus maybe the much more prevalent and much more kind of near-term utility that might be available from using human genetics for risk stratification, for augmenting a physician's toolkit in understanding a patient's risk of disease and taking preventive care measures. And so in a lot of those bases, I think the field kind of got obsessed with understanding the heritability of common diseases to some extent, and kind of I've been surprised at how long it's taken us to find clinical proof points of how that exact same data, now we're finally sort of talking about the idea of polygenic risk scores and how that might be used to guide preventive care for the right subsets of patients. And that's kind of been shocking to me, because in some sense it's the exact same analytics that we were obsessed with, more than 10 years ago, but in the context of chasing down the so-called heritability of a common disease.
Yeah, that's such an interesting point. I feel like I'd like to go back and see, because it feels like all the pieces were there. There were very big genome-wide association studies early on in common diseases. And if somebody had said, what if we just added up all the little effects and made a score? I imagine somebody did do that. Maybe it
they did. Yes.
Was it just ignored, or why is it 10 years late?
It's a good question. And I think I was really lucky to be a part of a lot of the groups that have now pioneered, really great publications and descriptions of the utility of polygenic risk scores clinically. And I think it was just at that time, the academic community kind of was in a quest to prove any utility of common disease variation. And honestly, that was such a raging debate, this common disease, rare disease debate, common disease, common variant, rare variant hypotheses. And
it's still a raging
debate.
It's still rage over it.
Yeah, I agree. But it was like there was almost this need to even justify pursuing more common variant profiling in the first place. And the most sort of fundamental question you could ask about utility at that time felt like it was heritability, which was to say, like, what fraction of a disease's causal, genetic architecture is tied up in common variants versus rare variants. And if we could just definitively answer that fundamental question, then we could decide whether we want to pursue more common, variant profiling in more people versus more rare variant profiling, in more selected patient populations or something like that. And that felt like the underpinning of a future research strategy to some extent. And I think people just didn't, it took a while to pose the very clinically actionable question of hey, is there a subset of breast cancer patients who actually have a lifetime risk of breast cancer that's comparable to that of a BRCA allele? And framing the question that way suddenly made it so much more enticing, I think, that now finally people are starting to pay attention. Well, hey, if I'm in that group,
I'd love to know that. Yeah.
Maybe I would act on it. Maybe my doctor would act on it. And at least the information, currency feels valuable.
Do you think there's any major barriers still stopping polygenic risk scoring and stratification from going through the healthcare system, besides the healthcare system itself and how it's structured to pay? Is that the major one, or are there others that you see?
I think there are lots. I think I alluded to one, which is kind of this subtle but important distinction between a clinical diagnostic and, an informative tool. Yeah. Holding polygenic risk scores to some kind of a binary diagnosis of yes or no, this patient is at high risk or not of having, say, diabetes or coronary artery disease doesn't really work. Because the scores aren't perfectly predictive. And, then you can kind of get into an endless debate about what the right threshold is and, whether you should reimburse for it. And that is sort of a different context. But a much more useful framing, I think, is to recognize that doctors have been using imperfect risk stratification for decades. The whole Framingham risk score is an imperfect way to pick out patients who might benefit from additional preventive measures to halt the progression of or delay the onset of a high-risk condition called coronary artery disease. And we're only okay at it in the status quo. And so if there were, an additional piece of information that would go into a whole other set of risk factors, and now this has been really well described in publications, out of St. Catharisen's group and lots of other groups now, suggesting that, yes, that information is additive on top of an already imperfect but still useful risk stratification tool that can guide the following actionable care management steps, I think that's a much better framing. And so I think we still have to figure out how to roll that out. Should it be rolled out as a diagnostic? Should it be rolled out as a population health surveillance tool? Is there a need for health economic studies to really justify the inclusion of this testing? People don't talk about it enough, but they're cheap, right? Like genotyping, whole genome-wide genotyping is dirt cheap at this point. You can do it for under $100. So to some extent, I think the fixation on health economic proof is a little misguided, because this is a one-time test. It's cheaper than, at this point, a lot of standard lab tests. And yet I do think practically that is something that we're going to have to do.
Absolutely. A couple episodes ago, I had Robert Greene from Harvard on, and the point that he made was one of the challenges from a reimbursement standpoint is how fragmented the healthcare system is in terms of who's actually paying for it. Because for you, it may be coronary artery disease. For me, it may be something completely different, but you've got a single test that is predicting, hundreds to thousands of different diseases. So figuring out how the economic healthcare system can actually wrap its brain around the fact that there's a single test that might, for you, deliver benefit to the balance sheet of one part of the healthcare system, but for me, it's someone else. But it seems like a solvable.
Well, and that's even more challenging for a test where the cost is still high. So if you think about, a high coverage whole genome, let's say, where exactly as you said, it could be used to determine your carrier status in the context of a young person planning a future pregnancy. It could be used to determine a polygenic risk score. It could also be used to determine your risk predisposition for rare, highly penetrant cancer predisposition alleles. It could just be used in so many ways that I agree. Then that question of kind of how do you parse the reimbursement for each of those applications becomes important. And the reimbursement matters because the cost of the test is still high. A high coverage whole genome is still in excess of, on the order of $1,000 anyway. This is really quite a bit cheaper. The concept of a common variant based risk stratification tool. We're really talking in the sub $100 already today. Effectively the cost our health system pays for routine blood work. But this would be a one-time test. So it does feel different on that basis.
Yeah, for sure.
But I agree.
No, definitely. Well, I'd maybe love to transition a little bit from polygenic scores, and we can definitely revisit this later into the future. But I want to talk a little bit about cancer.
Yeah.
Because you obviously spent a lot of time working on that at Flatiron. And from my perspective, and feel free to correct me if you see it differently, cancer seems very far ahead when we talk about personalized medicine. And I'm curious whether you agree with that. And if you do, why is that, and what's needed for everything else to kind of catch up to I looked at a recent graph that there are now clinical trials with 3 or more genetic biomarkers in cancer. More than half of all clinical trials now have a genetic biomarker in cancer. What is it going to take to get there in everything else?
Yeah. I think you're absolutely right. Oncology is at the forefront of kind of incorporating a precision medicine approach in routine care. And that's because cancer is the most genetic disease to some extent. Rare genetic diseases are, of course, the sort of flagship example of not only diseases entirely attributable to germline or, genetic variation, but, in that case, the mechanism is even more simple and straightforward. And so clearly, rare disease trials have had for a long time the requirement of a genetically confirmed diagnosis. Moving beyond rare diseases, when you look at diseases that affect large fractions of the population, some cancer types afflicting on the order of 10% of the population over the course of a lifetime, it is one of the most genetic diseases where the drivers of the disease are somatic genetic change that happens in a cancer cell to make it cancerous in the first place. And so I think it's just that core fact that we know it's the genetic transformation of a cell that led to the creation of the disease state that prompted people to focus so much on somatic tumor cell sequencing to understand drivers of the disease, and then subsequently build drug pipelines targeting those exact causal mutations. And the simplest explanation for why that profiling has become mainstream is that we actually have drugs to guide an action that's based on the testing, right? That started happening in the context of, maternity is kind of another example of where the NIPT test has become mainstream. Again, it's because that produces actionable information for a couple early in their pregnancy. I think a lot of people have argued, appropriately so, that even in oncology, our arsenal of targeted therapeutics is still not big enough to meaningfully change the course of a patient's cancer treatment in most cases. But even in the few cases where it absolutely does, that has been meaningful enough to make most patients and most oncologists, especially in certain subtypes like non-small cell lung cancer, recognize that it's totally imperative to be doing the tumor profiling to guide the use of targeted therapies. So the other piece is that in other common diseases, let's say diabetes, part of the reason that the adoption of a clinical diagnostic to guide treatment has been slow is that what we did learn from this decade of fixation on genetic architecture and heritability is that truly maybe a larger fraction of that disease's underpinning is in common variants. And so that means if you take 100 diabetics, they're much more likely.
They're all different.
No, they're much more likely to share, actually, their underlying drivers of disease because they're common. We're talking about a whole bunch of.
But they may be different. They may be different individual common variants, right?
Yeah, but we're talking about maybe thousands of variants, maybe millions of variants across the genome contributing to risk of diabetes, and many of them are at prevalence 30%, 35%, 50%. So as a result, in aggregate, the pathways contributing to diabetes across those 100 people are much more shared. Much more so than, let's say, 3 different subtypes of cystic fibrosis, each of which are attributable to a
completely different mutation. Yep, I'm with you there. Yep.
So, and so that's a good thing, right? That's why metformin maybe works in everybody with diabetes. And that's why many of our oral hypoglycemic agents are actually effective in a large swath of the type 2 diabetes population. So in some sense, like the failure of a type 2 diabetes and becoming a hyper personalized, care field is actually like it's a silver lining of the genetic architecture of the disease, because it means that the drugs that we produce might actually help everybody with the disease. But it also makes it harder to feel like we're giving people the best drug for them. Maybe there's not one.
Right. It's very productive. So I guess that it sort of implies that there's 2 broad groups. There's common diseases that are actually rare diseases masquerading as common. So, if we look at Parkinson's or ALS, there's monogenic subtypes, and those probably will have their own precision therapies. And then there's common diseases that are truly just common combinations, and those converge on a handful of pathways. And those might be much more challenging to develop precision medicines in the way that we think about them in the context of more monogenic diseases.
Yeah, and I'd even say maybe they don't converge on a handful of pathways. Maybe there are a lot of pathways involved, and that's what's making those diseases so complex. But those pathways, even if they are very numerous, may be largely shared across people with the disease, giving us the possibility to converge on a few therapies that work for everybody.
That makes perfect sense. And I'm curious how this insight that you've built over the last 10 to 12 years, from a genomics and precision medicine perspective feeds into your investment thesis, or if you have one of those, or view on the world. I wonder if you could just talk about how you evaluate new technologies, companies, therapeutics, and what you see at an early stage before the proof is really there in almost all cases that makes you take a bet that something could be the future.
We could talk forever about what makes an early stage life sciences company exciting to us. But, the core tenets for us are actually, we think a lot about the team who are the people working on behind this idea and working to make it reality, and how much does kind of their enthusiasm and expertise drive the likelihood of success for a novel technology. We look at how differentiated the core technologies are. In a biotech company, that could mean many things. It could be how differentiated is the assay that you've developed to uniquely profile patient samples, for example, and uncover novel potential disease targets. It could be how novel is the platform that you've built to optimize your design of novel antibodies. It could be more on the modality side of things versus on the biology target finding side of things. But, we do tend to, our approach at Andreessen Horowitz is to focus on a lot on how differentiated a technology platform is, even if that takes time to build. And it might take longer in some cases to build than a biotech company that's starting out the gate with potential assets in mind. But we think that sets the company up for kind of a very productive journey in which that platform can lay many eggs, so to speak, and make the company able to prosecute a very large number of targets over the course of its lifetime.
One of your investing focus points, I believe, is on unique data sets. I'm curious if you could talk a little bit more about that, whether it's genomic or other kinds of unique data sets that you're interested in.
Yeah. So we look at data sets, I would say, in largely two categories: data sets that drive discovery and data sets that drive development, clinical development of novel therapeutics. And in both cases, I think we're just seeing the tip of the iceberg in terms of how companies are going to creatively use data to accelerate and make more efficient both sides, discovery and development. On the discovery side, that could totally mean what we just talked about. It could be a company that's set up extraordinary access and know-how in mining large global population biobanks. It could be a company that's kind of crawling the UK biobank, all day, every day, looking to understand novel human genetically derived drug targets and prioritize them and look for sources of validation for them and be studying kind of careful subphenotypes within, populations where that drug target may be playing a role and so on. And that could be a really differentiated data approach on the discovery side of the house. It could be a company that's building an extraordinary experimental data set to nominate novel drug targets. One of the companies in our portfolio, called In Citro, is building a very unique platform oriented around using induced pluripotent stem cells, iPSC cells, to create controlled environments where the genetic perturbation can really be studied to say, well, if we make this genetic perturbation, what happens to the phenotype of the cell and how does that subsequently interact with the potential drug that could ameliorate the disease state. So there's lots of really exciting approaches happening on the discovery side. Maybe even in between kind of target discovery and development, there's a lot of data sets that are now being brought to bear on drug candidate discovery and optimization. So you could imagine having a data set that describes high throughput small molecule screens, or from, let's say, a DNA encoded library platform, and really building a data set that allows you to learn which features of a small molecule correlate with which functional perturbations of those molecules, or binding properties of those molecules. And you could have a really kind of unique data set that guides how you discover molecules. And then on the development side, lots of ways too, right? So, going back to the polygenic risk score discussion, lots of different data sets could provide really interesting ways of stratifying the population that you're hoping to treat. And in so doing, could enhance the effect size that you get to observe clinically of a candidate drug. So, I hope we'll see more clinical trials that are leveraging things like polygenic risk scores to determine who might be the right candidate, let's say, for a preventive therapy or even for a disease-modifying therapy.
Absolutely. Those are all great examples. Maybe this is a good opportunity to transition into the next 10 years. What are some of the areas that you're most excited about now and going forward? And in particular, any that are maybe off the beaten path that others aren't thinking about as much as you think they should be?
I'm particularly interested in exactly those. The framework that I use is sort of what I just outlined on companies that can step change how they do discovery as well as development. I think we've had a little bit of a period where we've sort of been skeptical of target discovery efforts. And some people have argued that, well, we're in a target-rich environment in some fields, maybe in oncology, where we know enough targets and it's been a problem of drugging them and finding the right modalities to drug them. And I think we believe in that. And a large number of the bets in our portfolio are focused on novel modalities to drug maybe biologically de-risked targets. I do think, kind of a set of, maybe dark horse companies that are going to come up and surprise folks are going to have built really key capabilities around target discovery and are going to, sort of go back to the biology drawing board and say, what are sets of targets that we just haven't done enough work to characterize and prioritize and nominate as novel, truly novel biology that we could intervene on. And so I'm a little, maybe a little bit of a controversial position vis-a-vis folks, honestly, who've looked at the trail of sort of target finding companies having really struggled to produce really novel targets. My view is some of those are actually going to succeed and that we need to support their growth over the next decade.
Sometimes it just needs time, right? It's not that it's a bad idea. It's just a little early.
Yeah, exactly. That could be, companies that are really investing in single cell sequencing approaches for target discovery. That could be companies that are investing in, studying the function of previously unstudied cell types. That could be companies looking at classes of targets related to RNA function or epigenetic function, and classes of proteins that we just haven't fully understood biologically.
Yeah, absolutely. What is it, do you think, that's holding that back primarily? Is it simply new technologies like single cell need to mature and get lower cost and start to go along, or is it the scale of data sets isn't quite there? What are the couple of things that, levers that you feel like could need to change by an order of magnitude in order for that transition to really happen in a big way?
I think the transition is already happening in the sense that I do see more and more efforts to expand the pool of novel drug targets. I think it just becomes a tough risk trade-off. And we have this conversation with companies that we work with focused on novel modalities all the time. Would you rather use your modality to tackle a target that has been universally agreed upon as a meaningful disease target, or would you rather take on kind of double stacked risk to pursue both a novel target and bring your novel modality to it? So I think it's not that target hunting has been unsuccessful. It's partially that we've also made so much rapid progress on novel modalities, right? We're able to design antibodies kind of in better ways and faster and more efficiently than ever before. We're able to pursue concepts like cell therapy. We're able to pursue concepts like target specific cell therapy in the context of, let's say, CAR T cells and so on. So in all those cases, the modalities have ramped up so quickly that class of companies has over the last few years been a little bit more oriented around how can I point that at a target that's biologically de-risked. And so I think we're now going to see a wave of companies that are going to say, maybe I will take on some stacked risk. And in some cases, I'll pursue my first couple of programs against de-risked targets. But then maybe my next ones, I'll pursue targets that are both novel and that may be also uniquely suited to my modality, where maybe another modality might not be able to exploit that biology the way I could. And that'll still be a way to demonstrate the power of a new modality.
How much of your you're investing kind of on a percentage basis is in, we've talked a lot actually about drug discovery and genetics and targets, but I know you have, you're still practicing doctor at Stanford. And so you have a unique perspective actually on the day-to-day patient care, which is in many ways really far, in terms of time and universe from drug discovery. How much time do you spend on the very early and, into clinical development versus innovations in day-to-day patient care?
Yeah, you're right. Sometimes the universes feel very far, but for me, it's really a source of tremendous personal inspiration to meet patients, every week who it's just very clear we don't have the right treatment paradigms for. I focus on cancer survivorship in my clinical practice. And so I see patients with late effects from cancer therapies. I see patients with, terrible graft versus host disease after a bone marrow transplant. I see patients who have lots of contexts in which our cancer therapies either didn't work or really created a lot of side effects that I wish we could have avoided somehow. And so, it's just always kind of humbling to remember that even though we're developing technology at what feels like such a fast pace sometimes, the patients in front of me haven't yet had the opportunity often to really access that or are still looking for more and deserve more. I will say some of my connectivity to care delivery does inform our thinking around ways in which development could be fundamentally changed. Our processes by which we educate patients about clinical trials, enroll them in clinical trials, follow them when they're on a clinical trial, learn about all their symptomatology while they're on the clinical trial are just behind. We're not bringing 2021 level technology to that problem just yet, and I think at the same exact time as those technologies feel like they should come online, patients are also changing their attitudes towards development. They really want to know about trials. They are eager to find out ways in which they could, I hope everybody, I've had so many patients say, I hope everybody who could possibly benefit from it will learn from my experience and what I've tried and what worked for me and what didn't. And so there's just that convergence of technology, provider willingness to adopt technology, and patient interest in being a part of research, I think is going to produce some real development advantages for our next generation therapeutics developers.
Absolutely. One of the things you kind of mentioned there was alluded to was the time period between, you can see Star Trek level things going on in early discovery and gene editing and gene therapies. And it takes 15 years sometimes to get actually into the healthcare system at a large scale. And sometimes it never gets to a large scale because it's so expensive. When you look at the whole system end to end, how do you think we get it down from, 15 years to something more like 2, 3, 4, 1 year months? If we look at, we haven't mentioned COVID all day, so, but I'll mention it now, that going from, viral sequence to vaccine in 2 days and through approval in a year. What do you think we need to do in the system as a whole to get everything down to a year like that?
Yeah, I do think, Operation Warp Speed and some of the ways in which government and private sector came together to accelerate vaccine development, not just vaccine development, also antibody development and other therapeutic development and also diagnostic test rollout, was extraordinary. And I hope there's no intellectual reason why that can't happen more broadly. There are there are kind of real technical challenges, let's say, in translating an mRNA vaccine platform into a platform that can be used to treat other diseases. 100%. There are real research challenges left and technical challenges left to solve that barrier. But in principle, in a case where we have a strong therapeutic hypothesis, we have a modality that can be spun up to treat it. I think antibodies are maybe an even more compelling example, right? Where We know how to make antibodies, but still historically the development cycle has been longer than it was for COVID antibodies. There's no reason why there's no hard barrier for why that can't repeat itself in other parts of our biotech industry. And so I really hope that, that we do get to see repeat examples of it, whether for other infectious disease pathogens where we're seeing a whole flurry of activity now, or for major disease therapies in other areas of unmet need. I think it will happen.
Do you think it's, is it likely to be like the Roger Bannister 4-minute mile where no one can run a mile and then as soon as someone does, everyone breaks it the next day? Do you think we're going to get to rapidly faster timelines because people see it's possible and have a playbook, basically?
I think so. I wouldn't underestimate some of the underlying platform technologies sort of just being ready for prime time at the moment that COVID called for them. And that platform development did take time. So going back to our discussion about, kind of being willing to put in the time to build a novel technology platform before it's ready to lay eggs, that's still going to be important. And I see a lot of platforms coming online, in the gene editing space and gene therapy and cell therapy, even new platforms in the antibody development world. We invested in a company called Big Hat Biosciences that's really bringing ML and protein sequence optimization to the antibody space. And so, that's also a platform that will take some time to develop. But once the platforms are ready, yes, absolutely. I think there's going to be a little bit of that. I hope there's going to be a little bit of that.
Sprinting.
Yeah, sprinting to the end now that we've seen that it could happen, right? Maybe you need to get the right shoes and, develop the cohort around you that's kind of cheering you on. And maybe you need to collaborate with another, maybe you need to team up, right? Maybe it'll be a relay race. And that's the best way to kind of collectively run the longest distance. But I think we've seen that happen. Companies have been collaborating a lot more than we historically saw. That could be a company that, is building a novel, developing a novel antibody that finds a really great collaboration with a manufacturing group that's optimizing that part of the equation. It could be a company that's working with a global CRO to really accelerate the rate at which patients can enroll in a clinical trial that they're running. It could be a gene editing company that provides cargo, but collaborates with another company that's making the delivery vector. And so these kinds of collaborations will help, I think, break some records, even if we break them by being part of a relay race.
I love that. Maybe that'll be the title. We'll see. One last question, because we're just running up against time here, and hopefully a quick one. We use the term personalized medicine all over the place, and I think everybody has a different personal vision or even definition for what that is. I'm curious what it looks like to you. What is personalized medicine, and how do we know when we get there?
Yeah. So my view on this one, actually, I think my PhD advisor, David Altschuler, said this at one point when sort of in frustration with the field, saying that the only way to get to precision medicine is like somehow to have everybody's therapy guided by their genome. He used to say, doctors have been doing personalized medicine for decades. Like, it's crazy to think that, to assert that doctors have not been providing care that's tailored to the patient they see in front of them, right? We've been diagnosing disease in individual people. We've been, prescribing lifestyle modifications that are specific to the patient that we see in front of us. And we've been working really hard to connect with people as individuals to influence their care. So in some sense, I do kind of share that view that we are already practicing precision medicine. And the sooner we can acknowledge that even our status quo has lots of components of imperfect, but still aspirationally precision care, that will actually make it easier for us to infuse genetic information and other more stereotypically precision medicine approaches into our standard of care, because it gives us a framework to understand that, hey, look, Every time a patient walks
Nothing
in,
new.
I'm already calculating scores for their future breast cancer risk and their coronary artery disease risk, and, looking at their medical history and asking which risk factors they have for what conditions. I've already been doing that. And so now if I can supercharge that with some additional genetic information, I think it's more attractive to me to think about a world where we make already precision medicine, more precise and even more impactful for patients, as compared to this sort of step change view, which is a little bit misguided, I think.
Absolutely. Yeah, I really like that. And great way to end. Thank you. If people want to follow you, I think you're Vineeta on Twitter, right? Which is an amazing Twitter handle. Well done with that one.
Thank you.
Thank you so much. I really appreciate it. It was a lot of fun. I learned an incredible amount, and I appreciate you taking the time.
Thanks so much. Thanks for having us.
Thank you for listening to this episode of the Genetics Podcast. If you enjoyed the podcast, we'd really appreciate if you left us a review on your favorite podcast player, or even better, you can tell a friend who you think might like it too. As always, you can reach us anytime at podcast@sanogenetics.com. We really love to hear from you all about any feedback you have, guests you'd like to hear from, or topics that you'd like to see us cover in the future. Thank you again for listening, and we'll see you next time.